Today is about classic genetics, how we understood how information was passed down between generations, but before modern advent. So, with all of today, you kind of have to pretend that it's a couple hundred years ago. You may know more about DNA than the scientists I'm going to discuss today, and that is fine. Just realize we would reach different conclusions than what the researchers did today because we know a lot more about DNA. We know about genetic engineering and things like that. So, in the past, I've asked my students to see if they can roll their tongue or if they have a widow's peak, if their hair comes to a peak. And this is the data that I've gotten from them over the years. With tongue rolling, out of 135 participants, about 75% could roll their tongue, 25% couldn't. Interesting, that's a very clean number. And with widow's peak, about 75% didn't have a widow's peak, but 25% did. What does this mean? What could be causing this pattern? What do you think? Well, there's got to be something genetically going on. These numbers are way too clean. It's always 3/4 one, 1/4 the other. That doesn't really happen in nature. If there wasn't anything regulating these traits, it would be much more sporadic, maybe 30/70. So something must be at play. To understand heredity, heredity is how information is passed down over generations. For example, if you look at this grumble of pugs, that is the term for a group of pugs, it's a grumble. They have a lot of different characteristics that vary between them. Most of them can keep their tongue in their mouth, this one cannot. Most of them are fawn, a couple are fully black, and there are actually some domino pugs as well. So this is what we mean by traits, characteristics that vary amongst individuals. For today, we need to understand, well, what is causing a trait and why do traits vary? Here's what we know so far. We know that DNA is a molecule that contains instructions for proteins. Proteins are what make up traits. Think about the pigments we looked at in photosynthesis. All of these different plants have a different color because their DNA is instructing them to make a different protein that produces a different pigment or a different color. We also know that traits vary because of meiosis. When we made gametes, sperm or egg, crossing over caused diversity and independent assortment caused diversity. So with this variation, humans are getting 2 copies of every gene. They're getting one from one parent, one from another. You get 1 chromosome 1 from dad, 1 chromosome 1 from mom. Now, those genes, though, can come in different flavors. Here's what I mean by that. You remember that chromosomes are homologous. So they have the exact same information. So here I have 3 individuals and the 2 chromosomes they have, and we're looking at the gene for eye color. So that's the information that's getting passed down. Well, that information can come in a variety of forms. We call these different forms alleles or different forms of a gene. If the gene is eye color, so that's my information, all of these are homologous, they have eye color information, but they can have different alleles. These 2 individuals have an allele for brown eyes, and that's why they have brown eyes. This individual has 2 alleles for blue eyes. So it's still the same information being carried down, it's just being told in a different way. Alleles are just different ways of saying that same information. So we know that a gene determines a trait, which is different alleles. So I have these genetic things as DNA, which determines a trait like hair color, and that hair color can vary amongst individuals. So we have genetic factors and physical factors. With genetics, the primary battle today is not difficult. It's just a lot of work and a lot of vocab words. There's a really good video that I think is great for looking at an introduction to these vocab words. I'll put the link in the ad puzzle in a moment. Feel free to watch it. It's not required. I just think it's really good at getting the vocab before I talk about it. So we have genetic factors and physical factors. We're going to call the genetic factors a genotype. That's just any chemical, mechanical thing that determines what physical traits an organism has. We're going to call the physical traits a phenotype. So genotype are the genetic factors, phenotype are the physical factors. Why are we using this vague language? Because this is a time before we fully understood what DNA was and how it worked. We knew there was some kind of chemical thing, but we didn't know what it was, so we called it a genotype. And we knew physical things were something, so we called them phenotypes. With genotypes and phenotypes, the phenotype we always represent with what it is. So if you're tall or short, your phenotype is tall or short. Genotype's a little more specific. We represent genotypes with letters. We can use uppercase letters or lowercase letters, and generally speaking, it's almost always 2 letters to represent a genotype. Why 2 letters? Because you have 1 allele from each parent. So with that eye color example I showed you before, brown eyes, I have 2 uppercase B's, or an uppercase B and a lowercase b. With blue eyes, I have 2 lowercase b's. Now, we're not doing uppercase and lowercase just for funsies. There is significance behind using an uppercase or a lowercase letter. And that significance is if a gene is something called dominant or called recessive. If a gene is dominant, that means it is always expressed whenever it's there. So we represent that with an uppercase letter. If I ever have an uppercase letter, it's present, it's expressed. I'm going to make that protein. With recessive genes, these are genes that are only expressed if the dominant gene is not there. So we represent it with a lowercase letter. Here's what I mean by this. If I have 2 lowercase b's, I express the recessive gene. But if I have a big B and a little b, since the dominant is there, it's always expressed. A common misconception people have is that if something's dominant, it's more common, like a lot of people have it. That couldn't be further from the truth. For example, having 5 fingers on your hand is recessive. It is recessive to have 5 fingers. It is dominant to have 6. Obviously, you probably don't know that many people with 6 fingers. It's not very common, but it is dominant. So dominant does not mean how much there is. It just means if it's there, it's expressed. If you are born with 6 fingers, your kids will have 6 fingers. So what's going to happen when we combine genes? Well, we have our 2 gametes, and we talked about this with meiosis, sperm and egg. When they combine, each gamete's going to carry 1 allele with it, and they'll combine for an organism with 2. Think back to meiosis. Each of our cells have 46 chromosomes, so we had to cut those into 23. So the sperm is 23 and the egg is 23, so that when they combine, we have an overall of 46. Alleles, it works the same way. I have 2 letters for a trait. Let's say 2 uppercase B's for baldness. Each sperm will only have 1 B. Here's how that'll work. So here I have 3 eggs and 3 sperm. If I combine this big B egg with this big B sperm, the zygote, or the fertilized egg, the embryo, is going to be big B, big B. Organism started with 2, gametes had 1, offspring has 2. Let's look here. If I have an X and an X, that means there'll be a baby girl. Sex chromosomes are XX for female-bodied people. And if I combine the X and the Y, that'll give me baby boy. Male-bodied people have XY for their sex chromosomes. Now, these genotypes can come in a variety of different combinations. If I have 2 of the same letters, so 2 uppercase letters or 2 lowercase letters, we call that homozygous. Homo is same, zygous is the zygote or the fertilized egg. So the fertilized egg has 2 of the same. And here's a homozygote. It's all white to help you remember it. When there's 1 of each letter, so I have a big B and a little b here, I call that heterozygous. Hetero different, zygous zygote. And here are some heterozygotes. They're half black, half white. That was a lot of vocab at once. Once you master this vocab, it's downhill from here. We're just crushing Punnett squares, and it's relatively easy, and students usually have a good time with this part of the class. So just to review the vocab word, genes, chemical factors that determine traits. They have the recipe for protein. Alleles are different versions of that. So if I have a gene for eye color, alleles could be blue eyes, green eyes, brown eyes. Genotype is the genetic makeup of a trait. We represent it with letters. The phenotype is the physical characteristic, and we just list out the physical trait. Yellow hair, brown hair, black hair. If a genotype is always expressed when present, we call it dominant. We represent it with a capital letter. If it's expressed only when there's no dominant allele, we call it recessive and represent it with a lowercase. If the 2 alleles are the same, we call them homozygous. They could be both dominant or both recessive. If they have 1 of each, we call it heterozygous. Gamete is a sex cell. It only has 1 allele. And a zygote is a fertilized egg. It has the 2. That was a lot of vocab. Again, if you watch that video link I put earlier in this Edpuzzle, very helpful in reviewing that vocab. So now that we have an understanding of the vocab, let's look at the science behind how we understand how information is inherited. A lot of it is built on the work of Gregory Mendel. Modern genetics began when in the mid-1800s in an abbey garden where a monk named Gregory Mendel documented inheritance in peas. Here's a background with Gregory Mendel. Gregory Mendel wanted to be a science teacher in his time. He wanted to be a biology teacher. Back in the day, well, and still today, you have to take exams to prove that you know biology and that you're prepared to be a teacher. He failed those exams. And here's why I tremendously respect Gregory Mendel. When presented with failure, he didn't give up. He was still determined to pursue his passion in science and found a way around the system. So instead of becoming an official biology teacher, he thought, well, how can I find the time and the money to do experiments? Well, monks have a lot of free time and they're paid and they live there. So what if I become a monk? Then I can just do a bunch of science, even though I didn't get into the program I wanted. So he did just that. He became a monk to pursue science. He was really interested in genetics. He used the experimental method. He collected very meticulous data and wanted to know the answer to these questions. He wanted to know why offspring look similar to their parents. You, for better or worse, resemble your parents. Why is that? How are traits passed down from one generation to another? What is the actual mechanism for that happening? And why is there this difference between organisms? There's a family resemblance, they're somewhat similar, but they're also somewhat different. What could explain that? So Gregory Mendel decided to use pea plants as his main focus for answering these questions. And he did this for the practicality of it, and he just really enjoyed growing peas. With pea plants, it's very easy to extract some sperm and egg and make sure that the plant you want to pollinate the other plant occurs. You can make sure that there's no crossbreeding, there's no sperm getting the wrong place or eggs getting the wrong place. Whatever you pollinate will have offspring from that source. So he found what he called true breeding plants, and that just means the purest form of a trait. We don't use this language anymore, but that's what they used back in the day. True breeding as in this is the whitest of the flowers and this is the purplest of the flowers. The first generation he bred, he called the parental generation. Hopefully you did that in your pedigrees. And all the generations after that, he called filial with a number. So this is the first generation. The next generation would be F2. He did this for a lot of different traits. He looked at color, roundness, where the flowers pointed. For today, I just want to focus on his most famous experiment, which dealt with the color of the plant. So he took his two true breeding pea plants. He had a pure purple one and a pure white one, and he bred them together. Take a moment and make a prediction. What color do you think their offspring will be? Well, let's see if you're right. To figure this out, we can assign alleles to each pea plant. So I'm going to say that the purple one is a big B, and I'm going to say the white one is a little b. Reminder of our vocab, these are homozygous. This is homozygous dominant because it has two uppercase letters, and this is homozygous recessive because it has two lowercase letters. To figure out what their offspring are, I can actually accurately predict this before I breed them together using something called a Punnett Square. Hopefully you're familiar with that from middle school. This is a Punnett Square. It was not made by Mendel. Otherwise, it'd be called a Mendel Square. It was named after a scientist called Punnett. Here's what it does. We can imagine what the allele for the sperm will be on one side and what the alleles for the eggs would be on the other side and combine them. This gives us every possible combination of alleles in the zygote. So I'm going to take the eggs here. Reminder that gametes only have one allele. Organism has two. Gamete has one. Those combine to get us an offspring with two. So I'm going to put a big B here and a big B here. For the sperm, little b here, little b here. Now to solve a Punnett square, you just drag down the top letters. So I put a big B here, big B here, big B here, big B here, and across the side letters, little b, little b, little b, little b. So here's what we get. 1/4 of the offspring will be big B, little b, heterozygous, one of each. 50% will be big B, little b. 75% will be 100% are going to be big B, little b. All of the offspring are heterozygous. So what does that mean? Well, thinking back to your prediction, maybe you thought they'd be all purple, maybe you thought they'd be all white, maybe you thought they'd be a light purple, like they would combine. Here's what Mendel got. 100% purple flowers in his F1 generation. That confused Mendel. What's the deal? I bred a purple thing and a white thing together, and now the white has disappeared. This doesn't make any sense. His hypothesis was that each organism had alleles and passed them down to their offspring. But this seemed to disprove his idea because the white trait appears to not be passed down. It's totally gone. Huh. If Mendel had stopped here, he would have never made the pivotal discovery in genetics that he did. But thankfully, Mendel saw the scientific method all the way through. He wanted to know what was going on in those hybrids, so he did what's called a test cross. He took two of those offspring and bred them together to see what would happen. We self-pollinated them. I'm going to represent them as both being heterozygous. They both have a big B and a little b. Why? Because we proved that in our last square. Let's see what happens if I breed two of them together. So I separate my alleles. Here's a big B, little b, and I solve my square. Here I'm going to have a big B, homozygous dominant. Here I have a heterozygote, and a homozygous recessive. Very different than what happened when the parents reproduced and they were all heterozygous. If I wanted to calculate the ratio of the genotypes, which you will do in these practice problems, I'm going to count how many out of 4 each genotype produced. So I have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So in this problem, I would say my ratio for the genotype is 1 to 2 to 1. I can calculate ratios for the phenotype as well. What are the physical traits? Well, I know 3 of these are going to be purple because the dominant allele is purple, and if the dominant letter is there, it's expressed. So anytime there's an uppercase letter, whatever phenotype that is, it's expressed. 3 out of the 4 have that, so I'm going to have 3 purple plants and 1 white. Interesting. So Mendel was concerned that, well, where did the white trait go? Because they were all purple in the last generation. He bred that generation together and the white reappeared. This was a confirmation of what Mendel was thinking. The trait didn't disappear. It was hiding. The dominant allele hid it and the white was actually recessive. So now he has a ratio of 3 to 1. The information didn't disappear. It was there the whole time. It just had to have a specific combination to reappear. Remember, dominant and recessive, something you probably already know, Mendel discovered. So this was our first discovery of things like dominance and recessiveness in traits. So just to review, he had 2 true breeding plants, a purple and a white. His offspring were all purple. What? That doesn't make any sense. Where did the white go? He bred those together and the white reappeared. What does this mean? Well, it means that some traits are dominant, that they will be expressed when present. Mendel proved that that's a phenomena with traits, and that it'll mask other alleles. In our example, the purple dominance masks the white color. This also shows that alleles can be recessive, that if it cannot work, if the dominant allele is not present, then that white will reappear and be expressed. He also proved something called the law of segregation. Let's look at that specifically. It says, alleles separate during gamete formation and combine during fertilization. I've been telling you this the whole time. Each individual has 2 alleles. They separate during sperm and egg formation. That's what we have in our square. We put 1 letter on the top, 1 letter on the side. They then recombine to 2 during fertilization. This is occurring because of meiosis, and that's why I taught you this first. In meiosis, we go diploid to haploid, 46 to 23. Mendel proved this without using a microscope. Just using the frequencies of his pea plants, he was able to show that the genetic material gets cut in half. Each offspring receives 1 from each parent, and they combine into a new frequency. Instead of calling it meiosis, he just called it the law of segregation, that the alleles are separating. This is happening during Metaphase I. That's when we have that first separation of alleles. Each individual starts with 2 copies of chromosome 1. During Metaphase I, each cell gets 1 of those chromosomes. The alleles have segregated. Pretty impressive for not knowing anything about DNA. Now, the crosses or Punnett squares that I just showed you are called monohybrid crosses because they're only related to 1 trait. We just looked at color and we did a 4-square Punnett square. Life is a lot more complex than just 1 trait. Oftentimes, we want to look at how two traits are inherited at the same time and do something called a dihybrid cross. Maybe I want to look at seed color and its shape instead of just looking at one. We can do this using Mendel's rules of inheritance, but it's going to be a little more complicated. Let's see what happens in this example. So I have a round yellow pea, and I'm going to breed it with a green wrinkled pea. Here's what I get for the F1 generation. Pretty similar to what Mendel got in his experiment, right? Only the dominant traits are expressed. So this tells me that yellow's dominant and round is dominant, and that green is recessive, wrinkled's recessive. Notice that I'm representing these genotypes with 2 letters instead of just 1. The Y's here represent the color, and the R's represent the shape. There are always 2 alleles for any trait. So since I have 2 alleles, I need 4 letters. 2 letters for the color and 2 letters for the shape. If I breed those hybrids together, I should expect 3 different combinations if it's going to work the same way as pea plants: homozygous dominant, heterozygous, and homozygous recessive. But in this case, you actually get 4. We're going to have 9 out of 16 be yellow round, 3 out of 16 be green round, 3 out of 16 be yellow wrinkled, and 1 be green and wrinkled. Interesting. That gives us a ratio of 9 to 3 to 3 to 1. And pro tip to make your life easy with these problems, anytime you're doing a dihybrid cross and you're crossing 2 heterozygotes, the ratio is always 9 to 3 to 3 to 1. That is the only possible combination if you are crossing heterozygous organisms. Now, there's something special going on here. Let's say I tried to solve this like a normal Punnett square. And I just did a 4 square. There are my alleles. I spread them out, and now I have 4 different combinations. So that means my ratio should be, if this is true, I should have 3/4 or 75% yellow and 25% green. Let's look at what I have here. That doesn't pan out. If you look here, I have 4 green. I also have yellow wrinkled. There's no yellow wrinkled here. Weird. So we can't solve these Punnett squares using just the 4 square. We experimentally prove that these have to be the results, but our square isn't matching it. And if theory doesn't match the world, scientists throw away the theory until we find something that matches the actual data. So to solve these kinds of problems, instead of doing a 4 square, we have to do a 16 square. Yay, so much more fun. If you distribute the traits and do 2 alleles for each gamete instead of just the 1 and complete that square, you will end up with the ratios that Mendel got in his pea plants. There we have the green wrinkled now. We have the yellow wrinkled. We have the green smooth. These predictions match the data. That's the key theme here, and that's the activity we're going to end today with. I'm going to give you data, and you have to show me theory that matches it. Data proves our theory. No predictions are worth anything if they don't match reality. So let's do a practice problem of a dihybrid cross. In this example, I have a tall green pea plant. I'm going to represent that as big T, big T, big G, big G. T's are tall and short. G's are green or white. Cross with a tall white pea plant. Big T, little little g, little g. I need to figure out what to put on the top of each of my squares. To do that, we're going to get acquainted with a familiar friend from the world of mathematics, foiling. Here's how we distribute the alleles. First thing I'm going to do is go outside, inside. So my first gamete is going to be a big T and a little g. After I go outside, inside, I'm going to go outside, outside. So now I have a big T and a little g. If you get lost on this, you have a copy of this in your assignments. Just always follow this arrow order. So I did outside, inside, outside, outside. Now I need to do inside, inside. So that's going to give me little t, little g. Then I do inside, outside, little t, little g, and now I have my 4 gametes. Your 16 square Punnett squares will only work if you foil before you set them up. So let's do that now for this top one. Same rules, outside, inside, outside, outside, inside, 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 outside. But since they're homozygous for all the traits, I already know it's going to be a big T, big G in each square. So now you just drag down your letters and drag them across and solve like you would a normal Punnett square. There are just a couple grammar rules I want you to be familiar with. So let's solve this first square here. I have two big T's, I have a big G, and a little g. Notice what I did. I kept the T's together and I kept the G's together. You always want to write the letters in the same order that they're given to you. It's just a grammatical rule of Mendelian genetics. So T's first, followed by G's. Notice also, I could have written little g, big G, but instead I put the big G first. That's another rule you always want to follow. Dominant alleles always are written first. So you keep your letters together, dominant first. So let's continue to solve this. I'm going to have the same combination, big T's, big G, little g, same combination, same combination. And I already filled out the second row because it's the same story. Now we want to fill out this square. I have a big T, big G, little g. I'm going to put my dominant alleles first, keep the T's with the T's and the G's with the G's. So I have big T, big G, little g. And that's how it ends up going for the rest of the square. Now you'll often be asked to calculate the ratio of each one. In the example I showed you before, since it was 2 heterozygotes, it was 9 to 3 to 3 to 1. Here, I'm going to first look at how many are tall and green. I'm tall if I have a big T, I'm green if I have a big G. All of them have at least one dominant letter, so all 16 are tall and green. This is what we call Mendel's law of independent assortment. Now, that's a word you've seen before, and for good reason. If you remember from meiosis, during Metaphase I, the chromosomes would line up randomly on the metaphase plate, and we called that independent assortment. It is the same thing as what Mendel discovered. Mendel discovered all of this without ever using a microscope and seeing. Because of that independent assortment, we can end up with 4 different combinations of alleles because they could end up on either side of the metaphase plate. This is why when you do your dihybrid cross, you have to foil. By foiling, going outside, inside, outside, outside, you are counting all the possible ways the chromosomes could be lined up on the metaphase plate. We call that independent assortment. Just to review, Mendel proved that traits can be dominant, expressed whenever they're present, or recessive, expressed when they're not present. How? The purple plants were dominant. The white disappeared when it was just purple, but the white reappeared in the absence of that purple allele. He also proved the law of segregation. I showed you this with a monohybrid cross, that for any single trait, the parent has 2 alleles, they separate when we form sperm and egg, and they combine in the zygote. A Punnett square represents that. The individual letters on the top are the gametes, and the 2 letters in the middle of the square are the zygote. We also saw the law of independent assortment. We have to foil when we do a dihybrid cross because of that independent assortment in meiosis. The chromosomes can be on either side of the metaphase plate. So by foiling, we calculate all the possibilities that can happen with that.